So sometimes when you're going through that process that you know already with helicase, right? Helicase splitting the DNA and polymerases adding complementary bases in opposite directions, every once in a while, a mistake is made. The wrong base gets added. And that can have various effects. Usually that problem gets fixed quick. You have several different ways of fixing mistakes in replication. You already know about P53. We talked about the 3 ways it works, where it can fix a mistake, it can produce a thing that fixes any mistake. It can stop the cell cycle until a mistake gets fixed, like keep a cell from dividing so it doesn't pass on the bad DNA. It can even cause a cell to self-destruct if it has a bad segment of DNA. But every once in a while something gets through. And so let's talk about sort of the 3 ways that can look like if it's what we call a point mutation. 3 different kinds of point mutations that we want to look at. Point meaning it's one base that's being changed. And basically, to put it simply, the 3 ways it can be changed is you can replace a base with a different one, you can add an extra base, or you can lose a base. When a base gets changed, when it gets replaced with another one, we call that a substitution mutation. As an example, we're going to use the DNA that we started with, minus the stuff we cut out of the mRNA. So we're going to say, for this mutation, that there's a G where there used to be an A. So it was an A, now it's a G. Draw that, and then get your secret decoder wheel out. And I want the before and after. I want what 3 amino acids we had before the mutation, and what 3 amino acids we have after the mutation. So what do we got? What was it before?
Methionine, proline, lysine.
That's what it was before?
Oh, and
Methionine. Yeah, methionine, leucine, lysine. Those are the ones you came up with initially. Now, those of you that are looking at me funny, the problem is this is DNA. That's not what you put in the wheel, right? You gotta, yeah, I know you did before, after, and after before. But you gotta transcribe it before you translate it, right? It's mRNA, so we gotta go, okay, this is GAG, that means it got transcribed into CUC, and that's what we put in the wheel. So it was leucine, and now it's proline. Good, good. Did the other 2 change? Yeah, doesn't touch those. There's still methionine here and lysine here. Great. So, all right. The other 2 kinds of point mutations are called, these are pretty easy to remember, oops, insertion and deletion, which is what it sounds like.
So before the mutation, it's put an A and a G.
It was an A. Now it's a G. That's what I was trying to say. Probably could have drawn it better. What does that say? Good. Dog has big hat. Awesome. Substitution mutation. What does it say now? Makes a little less sense. Dog was big hat, but still English. That's nice. Good, good, good. because you figured out right away that I was doing a 3-letter reading frame, right? That makes sense? That's pretty much what your cells are locked into, is that 3-letter reading frame. So an insertion mutation, for example, if we keep that 3-letter reading frame, what's that say now? Dorgasbegat. That's not English anymore. So that is just a dumb example so that you understand that these two are significantly more serious than substitution for the exact reason you just saw, that your cell uses a 3-letter reading frame, a 3 nitrogenous base at a time reading frame. If you add or subtract a nucleotide, it absolutely throws off the reading frame. So for example, same DNA, except now we've stuck a G in between that first T and A. Let's see what that does. Do the wheel thing. What 3 amino acids are we looking at now? Disturbing. What is it? What three amino acids?
Okay. Threonine, alanine, and glutamine.
Threonine, alanine, glutamine. Everybody agree? You do it right? I think you did. Yeah, because this will be A C U G C U C A A, and the C is just left over. Yeah. Basically, when one of these two happens in the much longer strand of or DNA than this, it's going to change every amino acid after downstream of the mutation, which in pretty much any case is going to totally screw up that protein. Not least of which would be because now the stop codon is not going to be where it's supposed to be, right? So these are mutations you often probably wouldn't see in a living thing because it doesn't result in a living thing. Because they're just not going to have that protein. It's probably something they need. Good. So, any questions about these 3 kinds of mutations and why they have different names and effects? Cool. That's what your homework's going to be. Let's mention a couple other things. Because beyond replication mistakes, the other thing that can cause DNA mutations are environmental things, which we call mutagens. So by far the most common of those is ultraviolet light, since we all hang out in the sun sometimes.
Grace Alexander, can you come down to the receptionist desk? Grace Alexander.
And UV light happens to be good at getting through cell and nuclear membranes and messing up DNA, which is generally fine, because the worst it can do is kill a bunch of cells, which is what happens when you get a sunburn. And then a few days later, you're peeling. Well, that's the dead skin you killed with ultraviolet light. Except there is that one change that can happen to your DNA that is a problem, right? Because it only takes one cell to forget how to stop dividing to cause skin cancer. So most of the mutagens we pay attention to are more specifically carcinogens. They're the kind that can result in that one change that causes cancer. So UV light, various other kinds of radiation, any tiny particles that can get into your lungs. It's not just smoke. Woodworkers have to worry about it because of sawdust. Asbestos is the same deal. It's not naturally toxic. It's just tiny particles that can get in your lungs. And anything like that can mess with your lung tissue and cause cancer. So any mutation, regardless of what it is, can basically have 3 different effects. The first is going to surprise you, or at least it surprised me, because there's a number in there that kind of blows my mind. By far, the most common effect of a mutation is nothing. Most mutations have no effect on a living thing. And that is because, and this is the number that really kills me, a full 97% of your DNA doesn't code for proteins. So having a mutation there is not going to affect a protein. If you're wondering how that can be true, I can get you to 50% real easy. You already, somewhere in there, know that 50% of your DNA is non-coding, because you know how transcription works. RNA polymerase splits the DNA and then matches bases to one strand, not the other. So for every gene, there's half of the DNA that's not being used. So a mutation there is fine. Right? So there's 50%. Then add in the fact that you've got that third wobble base in a lot of cases, like on your wheel, right? Like between 5 and 6 o'clock on your wheel, arginine. As long as you get the C and the G right, doesn't matter what's in the third spot. But more importantly, that genes are spaced out with usually thousands and thousands of nucleotides in between. There's a lot of spacer DNA that we kind of didn't know what to make of at first. We'll talk about what it does on Monday. It turns out it is not just junk DNA like we thought it was. So no effect is the most common. It is possible to have helpful mutations, which sounds weird and very X-Men. But shouldn't. Because you've learned at least a little bit about evolution and natural selection, right? Where do you think new traits come from? This is it. Evolution by natural selection doesn't work without helpful mutations. You, in a very real sense, are a big collection of all the mutations that worked. You're the success story. Everything you got started out as a mutation at one point in your ancestors, and it helped them because they successfully reproduced. And then, of course, there's bad things. Chapter 14 gave us an interesting example of sort of a gray area between 2 and 3, right? The idea that, okay, there are genetic disorders that are mutations that can kill you if you have both copies of them. Or if you only have one copy, they can help you, right? Having one copy, you don't get malaria. Having 2, you've got sickle cell. Sort of depends, right? So those are the options, basically. Monday, we'll talk about that 97% of DNA that doesn't code and how it's really not just nothing. We're learning more and more about it every year. And we're getting close to the end. So test should be Wednesday, Thursday at the latest. Well, I don't know whether the grade will go on this quarter or next, but I think we'll get the test done at least. Right. You might have some say in whether it goes on this quarter or next. What's your instinct?
No.
Next?
Yeah.
I don't know. Maybe we'll vote. All right, I have something for you to practice with mutations and to practice. Some of you need practice with the wheel still. You get, listen, you get a length of DNA to start with that you have to transcribe and translate with the wheel. Then they start to mess with it in various ways. In other words, different kinds of mutations that you have to analyze and figure out what to do. Good practice. This is due Monday. Sorry, due Monday.